All right, this is video for question 1 of 3, parts A, B, and C. I will be reading the questions off my computer, just reading the questions before I answer them, but I will just be using what is on the sheet to answer them. I'm taking this on my iPhone here. But yeah, let's get into it. So for problem 1, our question is, proteins are polymers made of amino acids. Draw the structure of the dipeptide THR-LEU and label the peptide bond N terminus and C terminus. So I answered this by the following. So proteins are polymers made of amino acids. A dipeptide is formed by linking 2 amino acids, and a peptide bond forms between the carboxyl group of one amino acid and the amino group of another. Here's a simplified representation of the dipeptide. THR-LEU, the peptide bond is between the carbon C equal to O and the nitrogen NH. So that shows that there. So it answers part A. Moving to part B, our question here is, protein structure can be classified as primary, 1 degree, secondary, 2 degree, tertiary, 3 degree, and quaternary, 4 degree. Explain what types of forces stabilize each of these structural levels in your explanation. Be sure to point out how each of these structural levels are different. So here I broke this down into 4 different parts. First being primary structure, 1 degree, which is the sequence of amino acids stabilized by peptide bonds. Our second is secondary structure, 2 degree, localized folding patterns like alpha helix and beta sheets stabilized by hydrogen bonds. Our third, tertiary structure, 3 degree, overall 3D structure of a single polypeptide chain stabilized by hydrophobic interactions, disulfide bonds, hydrogen bonds, and ionic bonds. Finally, our fourth, quaternary structure, 4 degree, arrangement of multiple polypeptide chains stabilized by the same forces as tertiary structure. And that is all there. Now moving to C, our question is, enzymes are protein catalysts that increase reaction rates by lowering the activation energy. Describe 2 types of enzyme substrate interactions that lower activation energy. You may wish to include drawings in your explanation. So I answered this with 2 different parts within part C. So the first is lock and key model. The enzyme's active site is precisely shaped to fit the substrate like a lock and key. This ensures specificity in substrate binding. Our second part to it is induced fit model. The active site undergoes conformational changes upon substrate binding, providing a better fit. This model accounts for flexibility in substrate recognition. And that is just the 2 different ones there, or the 2 different types of enzyme substrates interactions. So that answers parts A, B, and C of question 1.